First row, front row. Okay. Is everybody sitting next to their friends? Good. We're going to do something different today. Uh, I want actually 3 rows. For something I want you guys to do, I want to have 3 rows. 4 rows, whatever. Everybody has to be in a row. Okay. And we need equal number of people in each row. So we've got 8 people. Good job. Perfect. Let's get started. 3 rows, right? We've got 3 rows, 8 in each. Good. We're going to play a game. Is everybody ready? These are called team building exercises. Today morning, I spent an hour looking at team building exercises for you guys because I want you guys to know each other's names. Seems like you don't even know each other's names, right? Okay. I'm going to do very simple things first. You'll start on the left and we'll end on the right. Okay? All you have to do is you'll say your name. You look at me, say your name. Once you're done, you'll look at then you'll just move your head to your left. He'll say his name, okay? Then he'll move his left head to the left. You'll say your name like that, okay? So we go like this in a row. What? I'm going to time you guys. We're going to start here, end there. And when he's done when he's looking at the wall, okay? Then your time ends. And everybody is going to go fast. We'll see how fast you can go. Okay? All right? We're going to repeat this again in a different way. Okay. All right. I have a timer. Ready? Okay? No cheating. Okay? Everybody's going to get started here, there. I have the timer here. I have a stopwatch here. I'm going to start the stopwatch. When I say start, you go. Cool? And let's see if everybody remembers their name by then. You're going to be missing out for the first one. Is that good? Did everybody get it? Okay, 3, 2, 1. 6.86. All right, let's see if you guys can beat it. Now, you don't look at her first. You look at me first. Tell me your name. Then you look at her. Then as soon as you look at her, she passes, right? Good? All right. Is anybody keeping a record? The first row is 6.86. Next one. 3. Oh, wait. 3, 2, 1, go. 5.87. They beat you by 1 second. Oh my God. Third one. Is it fun? It's a boring fun. You guys remember each person's name? I'm going to make you do this until you remember each other's names. Okay. 3, 2, 1, go. 4.5. Great. Yay. Now we're going to do something fun, different. You guys want to do the same thing again? The first row wants to beat the last row? Okay, let's do it again then. We'll do this one more round till I think of something else. Okay. Looks like we have people coming at 10:20. These people are from some class. Huh? Okay. Let's have fun. Okay, guys who just came, we're playing a game. You guys want to play a game? This is an epigenetics class for fun. I want people sitting in a row. You guys see? No. Sit in a row. Like all rows should be filled. Come on up front. Come on. It's okay. Come on. You guys are coming from some class, right? When do you have to go back? What time is return to class? Okay. I want 8 people in each row, or 10 if people want to sit here. 8 in each row? Got this? 8 in each row? Only one row has, or 2 rows have 6 people in each, right? For those 2 rows which is close to the door, or one row, go 2 people go this way. I want 8 people in each row. Okay. 8 people in each row. Do we have it ready? That 4th row is empty. You're going to play with us? Okay. Okay, can one of you sit next to the guy who is with the cap? Sorry, आपका नाम क्या है? Rishi. Rishi जी के next में कोई बैठेगा एक? Or it's okay, I think it's fine. Right? We're 8? Are we 8 in each row? Can you guys count? My university students, can you guys count? 8 in each row only. Okay, we have 8 in each row. Which row does not have 8 people? You have 10. You guys want to go back to the row where there is 6 people? Just for fun, you can come back up later. Okay. Which row does not have 8 people? Okay. Well, these 2 people, will you be 8? No, not still. Right? Or is it? I have a parallax error. Okay. All right. What are we doing? Can somebody explain from the front 3 rows? One person get up and tell the rest of the class.
Then I will look towards Prachi ma'am, and then she'll say her name, and then अपना ID. और जो सबसे पहले complete करेगा.
Okay, each row is competing. Okay, we're minimizing the time. आप लोग को English समझ में आती है, right? सबको. I mean, university students. I expect this. Okay, get this right. You guys want to start again? Let's start from the beginning. Okay, we'll see who wins. One round because there are so many people now. All right. And you guys can observe the first rows because they played it once. We're just having fun. Okay? I just want you guys to know each other's names. That's all. Right? The objective is for people to know each other. This is team building exercise. Okay? In a team, when we recruit people, this is what we do. We make them dance, we make them laugh, we make them sing, we make them call their names. Okay. The next time we'll do something else. So we'll do this first round and let's see how fast you guys go. Okay. All right. Start. Wait. I'm sorry. 3, 2, 1, go. Okay, 5.11. That was 5 seconds. Okay, 5.11. Let's see if you guys can beat them. Right? Team 1, Team 2, Team 3, Team 4, Team 5, Team 6, Team 7, Team 8. We have 8 teams. All right, restart. 3, 2, 1, go. Okay, that was 4 seconds, I think. Sorry, messed that up a little bit, but it was 4 seconds. Good. Or can somebody watch this? Computer? Okay, yeah, I need one of the timers. Why am I doing all the work? Okay. 3, wait. Stop. Restart. 3, 2, 1, go. Okay, 4.2. I think her second was 4.1 or something. So still, second row's winning. Let's see if the fourth row can beat the first ones. You guys are new to this, right? Which class are you guys coming from? Plant biotech? You guys are undergrads? Okay. 3, 2, 1, go. Okay, 5.35. Okay, redo this. Next one. Oh, hold on. When I say go. 3, 2, 1, go. 4.46. Okay, good. You remember your numbers. Restart. Okay. You ready? 3, 2, 1, go. 5.04. Okay, not bad. Who's that now? Yes. 3, 2, 1, go. 3.96. Awesome. They have 8 people, right? Okay. All right, no cheating. Last one. Let's see if you guys can beat 3.96. Okay? 3, 2, 1, go. Whoa, 3.2. He's just not doing the timing right. Okay, all right. Good. Done. Who wants to beat 3.2? Okay, my 3 rows up in front wants to beat. Okay, only those people who are willing to play will play. Who wants to play again? Because this is a very fast game, right? Okay, only the first 3 rows. The new younger generation doesn't want to play. The bachelors. Okay. All right, now I'm going to time because I don't trust him. All right. 3, 2, 1, go. Okay, 3.43. They almost came. Good job. Let's see. See how this becomes a better and better? They started with 6.87, the first row. Now they're 3.43. They did reduce it by 50%. How cool is that? So when people are given a challenge, they perform. That's why I'm hard on my people. All right, let's see if you can beat 3.2, guys. 3, 2, 1, go. Okay, 3.09. There we go. Yay. Okay, last third row. Let's see. So this guy was not really cheating, actually. 3, 2, 1, go. 3.5. All right, anybody else wants to play? That row which was 3.2 wants to do it again? Okay, you want to play again? All right. I just want to make people play who wants to play. I don't want to make everybody play. If they don't care about it, this tells me who's competitive here. That guy's competitive. He rose his hand twice. He's like, yeah, I want to do it. I want to beat Salil. Okay? All right. 3, 2, 1, go. 3.6. Where you guys? Oh, are they have 10 people? No. Oh. Okay, we'll do it again with 8 only. Who wants to? He wants to volunteer out. First row or the last row? Only one of them. Huh? You guys have 10, right? Oh, okay. Let's see. Do it again with only 8 people. So we'll start there this time. Oh, no, actually, let's start here because I want to do that again afterwards. Okay. You're going to go and you two are not playing. Okay. So when she's done, that's done. The Gyan people will kill me. 3, 2, 1, go. 2.74. Yay. All right, we're done. You know what? This game is very common in team building exercises. And when they do it, people have done it in less than a second. Well, it depends on how many people are there. So, right? That goes that way. Now we're going to do otherwise. Last round. Okay? We're going to go here and start there. Why does that matter? Because you guys followed a pattern. You're used to it. Your body's used to it. We're going to try to do it otherwise and see if this becomes worse. Okay? Right? This is last round and then we'll do epigenetics, guys. I want them to pay my tickets price. Otherwise, they'll say you didn't really teach anything. Okay. 3, 2, 1, go. 3.58, 2.74. What did you guys do last? 3.43, right? Okay, so you didn't do well as well. Right? This happens. Your body's tuned to it, right? Because you're tuned to responding quickly. Okay, next one. 3, 2, 1, go. 3.27. Is your name Kapil or Kapil Dev? The whole thing is Kapil Dev. Your last name is Dev, right? You're only supposed to have first name. You're letting your team down. I'm sorry, kidding. Yeah, it's fine. But Kapil Dev is a great name, man. You have a lot of responsibility for the nation. All right. Ready? 3, 2, 1, go. 3.23. Okay. This the row 2 and 3 do worse or are better than the last time? You did better than last time. Okay. Both of you did better than last time when you were going this way. So this person is a problem, you mean? Okay. Ready? 3, 2. Oh, where? Sorry. Do you guys have 10 people or 8 people? Who's the 9th one? Acha. Aap mat keliye, please. Sorry. I know you are the one recording me. Okay. You can delete this portion for sure. 3, 2, 1, go. Okay, 3.5 after a little bit of this. Okay. All right. Which one? You guys or the one behind you? 10 people. So you two are not playing. Sorry. Or you who can volunteer to not play. Who has the longer name? Those people should not play. That's a key, right? Be smart. You're not playing. Okay, so you were starting at you. 3, 2, 1, go. 3.6. So what was the best name, best so far in this round? You guys remember your numbers, right? 3.23, right? Third row. Okay, all right. Which one is that now? You guys, are you 10 or 8? Okay, so who was not playing? Last time who did not play? You did not play, so you're playing this time. So we are not, you two are not playing. Okay, 3, 2, 1, go. Did you guys really do it right? 3.22. Okay. You didn't cheat, right? Nobody cheated, no? I can't see everyone because you don't have to move your head until you finish your name, right? Don't go crazy. Okay, next this one. You, right? 3, 2, 1, go. 3.69. I like it. Everybody's in threes. And I don't have anybody in twos yet. The last time, who was in two? 2 point something? Okay. We're starting there, right? Okay. I can't hear anybody of your names. Everybody else should pay attention to their names. That's the purpose. Okay. 3, 2, 1, go. Did we do it or no? I didn't see. Did you guys finish? 3.7 then. Okay. Because I stopped at some point. That was 3.7. Okay, last one, right? No. Two more. Is that the last? Oh no, you only four of you. Who didn't play last time? In these two rows, you guys didn't play, or you guys didn't play, right? Okay. Well, that would be too bad. But it's okay. You guys missed out because you're late. No problem. You miss out things if you're late. Just koya or soya or koya, whatever, right? Okay. 2.73, right? The record. Awesome. Great. Now we're going to do epigenetics. Okay. Let's start. Oh, what is that? Okay, what did we do yesterday? We were here, right? I will keep showing you this picture again and again. What is this? Who can tell me? Epigenome map. It's a chromatin state map. I don't think half of the class understands it because they were not here yesterday, right? Okay, we're not going to talk about it right now. Because if I go on to this will take time. We can come back to this later. What are we going to talk about is, I think we didn't finish yesterday. We were talking about looking at chromatin marks, right? Epigenome mapping. But epigenome is important for what? Epigenetics is important for what? Huh? Specificity of gene expression. So we've been studying gene expression for last 3 days, right? Okay, how do you study gene expression experimentally is the question. Okay, so we're trying to study genes transcription or mRNA expression transcriptionally. Okay, so how do you do that? Who knows? Does anybody know how do you study mRNA transcription experimentally? No? Okay, so we make RNA. Remember, everybody knows this. We're going to recap this one slide for people who are new. I knew that people are going to come late to this class and people who are not here. So this is about transcription, right? There is a DNA. There is a transcription factor. There is an RNA polymerase. RNA polymerase is driving it. The one in the red is RNA. So we are making RNA. That's gene transcription, right? Most of you know this. Most of you understand it. People who came just for the game, do they understand this? Because you guys they're not here, right? For last few classes. How are you? So you're just randomly attending. Do you get anything when you randomly attend? Some things. It's okay. I like people to speak, okay? I'm a social animal. I want people to speak to me. Can you guys speak to me? Like the people in the back rows. Do you guys understand transcription? How many of you understand transcription? Raise your hand. I said, you said, I heard yes, sir, but I don't hear any Hands should be like this, not this. Okay, this, because I'm blind, I can't hear, I can't see. This. Okay, good. So only few of you understand gene transcription, right? Is that correct? Genes transcription is what? Somebody wants to say from the top 3 rows, because I see you guys all the time. So front 3 rows, just explain to your rest of your friends what is gene transcription in like 2 lines. Somebody, yeah, please go ahead.
Multiple factors about transcription factors. And then they recruit RNA polymorphism for polymerizing the mRNA, and the mRNA is then produced.
Thank you. Sorry, what's your name again? Soumya. Thank you, Soumya. So people get it? You take DNA, you make RNA through an RNA polymerase. That's called transcription for very simplicity purposes, right? But you want to study which cells are expressing which mRNA, that is which genes, right? So we want to study this. How would we do it? Let's say we have a gene of interest, X, right? Any gene of interest. We want to look at the quantitative levels of that gene. How many mRNA copies was made from gene X? I'm Elon Musk's fan, so I'll call everything X, right? Gene X. Okay, so for that, what you use? Something called quantitative real-time PCR. Really? How many people have heard of this? Very good. Many of you have heard of it. Where have you heard this from? Which class? What class? Recombinant DNA technology. Can you guys speak in one sur? If I was a singer, I'll make you sing, and you had to sing in one sur with me, right? Right? You guys go to concerts, no? You guys hear concerts on YouTube? And the guy singing, oops, let's sing with me, right? Okay. I was going to sing a song. Okay, let's see. Quantitative PCR, right? Yeah, I want to hear from you guys, right? What do you know first? What do you guys know? You've heard of this in your recombinant DNA technology class. Okay, who teaches that? Huh? Rajat sir. Okay. All right. One of you want to stand up, not from first 3 rows, and tell me what do you know about quantitative PCR? In my class, I make my students teach me. Please. No? Nobody? Just stand up, guys. It's okay. I'm not gonna bite. I'm not a dog. I'm just gonna tell you whether it's correct or not, right? Not in the first 3 rows, but I want this. Come on, an answer. I'm gonna randomly pick otherwise. There are no leaders in this class? Who understands? Okay, raise your hand if you know what is quantitative PCR, so I can pick a hand. Now nobody understands it. Oh, you do. Okay, come on. Let's go. Very good. Do you know how is it done? How is it done? Okay. I will leave it on Mr. Dr. Rajat, right? Rajat, to explain this to you guys. I was going to play a video, but I'll save my time. Okay, thank you. Quantitative PCR is nothing but a PCR with a quantitative measure to it. How do you quantitate something? Through measuring a signal that it is proportional to the mRNA quantities, right? That would be quantitative PCR, right? So proportional signals come from things like fluorescent probes, right? So these things use fluorescent probes. These technologies are made by smart people who know what they're doing, right? Okay, so in quantitative real-time PCR, you put your NTPs that are quantitative in nature, or you use a probe that has a fluorescent probes to it. So every time the qPCR happens, a certain amount of emission of fluorescent spectra comes out. The machine reads it and says, okay, this is what we got. Here is how many cycles I took to make 100 copies. The lower the CT, that is the cycle number, the more higher the expression, right? That is what is represented here by these graphs, right? So the red means higher number of copies, blue means second highest, and the yellow means third highest, right? Do you guys get this? Am I saying something wrong? Or is it reverse? Is it reverse? How many of you think lower CTs means higher copy numbers? What is the CT value? The cycle number. CT is just cycle number of amplification. How many of you think lower CT value, that means lower number of cycles, means higher number of mRNA copies? Okay, how many of you? Raise your hand again, guys. Be confident. Either it's a yes or a no. Yes or a no. And how many of you think lower CT equals to lower mRNA copies? Okay. What do rest of you think? Then it looks like only 20% of people think this, 20% of this they think. There's 60% of people seems like they don't know what's going on. I thought you guys said you have had this in your class, right? Again, I'll let you vote again. There's not general election. You don't get to vote only once. You get to vote twice. How many of you think lower CT values equals to higher mRNA number of copies? Okay, thank you. You don't raise your hand again. How many of you think lower CT values equals to lower mRNA copies? Okay, we have more people who think the second is true versus the first one. Is that correct? And some of you don't even think anything. Guys, come on, you are in a biotechnology class, right? Biotechnology, plant biotechnology, forensic science, whatever your stream is. You have had your recombinant DNA technology class, right? How many of you have taken recombinant DNA technology class? You can't lie on this. I have taken mine. So you don't know the answer to it because a lot of people are raising hands, most of you, but you don't know the answer to this. How many of you think you don't know the answer to this? Okay, now that's a lot of you. Okay, so the statement I made the first time is correct, right? Why? Because the higher number of DNA or mRNA copies we have, the more transcription I will have, because if I have 10 copies, I'll start at 10 different templates. If I have one, then I will start at one template, right? For me to go to 1 to 10 itself is the difference that is between the red and the yellow. Did you get it? No? Are you sure? What if I said what the statements I made just now are all wrong? How many of you can challenge me? Challenge me, somebody challenge me that all the statements I just made in last 30 seconds are wrong. And I was just playing a game with you guys because I like to play games, right? You guys are confused. Most of you are confused. All the statements I made were true. None of them were wrong. And I'm surprised that you guys even think that can be the case. If you start with 10 copies versus 1, right? You have 10 templates to begin with. And you have one template to begin with, right? It's a 10th grade math, guys. Or 11, like it's a 5th grade math. My daughter can do this. Oh, 10 copies, 1 copy. I'm going to amplify 10 copies. I'm going to amplify them faster, right? To a same level. Compared to when I start with 1, for the same level, I'm going to take longer. Isn't it true? The longer means more cycle numbers. So, more cycle numbers means less copies, guys, less. 1 is less. 10 is more. This is a 2nd grade math. You guys are undergrad. This is disappointing me. Okay? Seriously, I'm not kidding. I might be smiling, but I'm actually disappointed. Okay, this is the most sensitive method to determine the level of a gene expression. Now, I hope you got it. Go on and watch a lot of YouTube videos on qPCR, because I did have a qPCR video playing on my laptop because I expected that you guys don't know this. That's a shame. Literally, right? Okay, but I want you guys to go watch a YouTube video on quantitative PCR, please. All right, so that your concept is clear, you're not confused. 99% of you are confused. That's unfortunate. Okay, we can't have that in a undergrad level central university classroom. Okay, take it for what it's worth. I mean, okay, but you can't be confused about these things, guys. Not allowed. Can't do biotechnology if you can't figure this out. Okay, all right, simple gene expression analysis, right? Very sensitive methods. It tells you exactly how many copies you can have, because you can use a standard. If you knew I had X amount of mRNA, I can use that as standard. I can dilute it 10 times fold every single time, and I can run as a standard, so I would know how many are there in my experimental setup or in my cells. How many of you got what I just said in last 10 seconds? You got it? Sure? How many of you did not get what I just said? Okay, a lot of you did not get what I just said. Because I speak bad English? Because you don't understand the meaning of standard. Is that true? Okay. What does a standard mean anywhere? What does the word standard mean? In English, Oxford Dictionary. Predefined something, standard, right? The standard here is that everybody should have gotten 90% of the marks in their 12th grade, right? The standard is something where you know what you are looking at. Okay, a standard is important so that you can prime the machine to say, I've got 100 copies here, I have 10 copies here, I have 10,000 copies here, and if I use those different dilutions of a given reagent, if you will, in this case mRNA, okay, then I'm going to get certain CT values as I go along. I'll make my curve like that, okay, and then I will use an experimental mRNA, and I will plot that curve there and say, okay, here is how many copies do I have based on my standard. Now, how many people got this? Okay, is it better than last time? How many people still don't get it? It's okay, raise your hand, no problem. And I won't be mean, okay? But basically, what you're seeing on the screen right now is 3 different dilutions resulting in 3 different CT values, okay? The CT values are plotted here, and mRNA copies are plotted here, for example, okay? So you've got this no, that's not true. A fluorescent is plotted against the CT values, okay? All right. So basically, you can use a standard, let's say 10 to the power 5, 10 to the power 4, 10 to the power 3, 10 to the power 2, 10 to the power 1. That's 10-fold dilution, okay? Is that cool? Yes. Okay, you use that, and then you say, my experimental value is right here. I have a graph here. Let's assume a graph here between the blue and the red, okay? So imagine, oh, I'm sorry. Imagine that, what did I say? This was 10 to the power 5, 4, 3, something like that. So that value will be somewhere between 10 to the power 5 and 10 to the power 4, right? Did people get it? Yes. Oh, they were still playing name games. You guys got it or no? Okay, so the quantitative real-time PCR is where you use fluorescent probes to measure the amount of mRNA. You can use a standard, and then you can use your experimental setup, and you say, in my experimental setup, I got X amount of X copies of mRNA, okay? Of Y mRNA, or N number of X cop, N number of gene X, okay? That's what we are going to, right? Is that cool? Okay. You get it? How many of you really don't get it? Seriously, I'll try to explain it again, because this is such a simpler thing. No? No? You still don't get it? No? I need an answer, yes or no? This is just my first slide, guys. I have like 50 more. Okay. All right. Same way, you can look at a reporter where if you want to look at the. Okay. Let's go back. So that was if I had a probe against a certain gene, my gene X. Okay. Let's assume that I want to know what is the number, what is a certain promoter of a gene. Did you hear about promoter? How many of you remember promoter? Okay, good. So how good is the promoter? How much number is it firing, right? You can essentially use a separate method to say, okay, I'm gonna, rather than my gene of interest, okay, I want to understand how many, sorry, this touch is a bad thing, okay. Okay, the highlight, the heading of this slide is wrong, so don't look at that, okay? Sorry about that. This slide is meant for telling you if you want to assess activity of a promoter. Okay, so a promoter is how strong a particular mRNA, a promoter is, for example. That is usually used for things where you want to look at the activity of a specific transcription factor or whatever. This is used for screening a lot of things when you want to assess whether what factors modulate the expression of that promoter or the gene promoter, okay, of that gene. So gene has a promoter, right? So what you would do is you would put your, instead of your gene of interest, you would clone it. Do you guys understand cloning? Okay, you'll clone a GFP or a fluorescent promoter, if you will. Okay, when I say if you will, it means imagine. Okay, that's an American phrase people use a lot. This is a fluorescent probe, which is fused under a gene promoter. So it's just replacing your gene, because for my gene, assaying my gene, I don't have a way to assess it. But with the fluorescence, I can assess it, right? The hypothesis here is that whatever gene sequence here is does not dictate what is going to be the level of promoter activity. Okay, so I can replace the gene sequence with my fluorescent probe. Get it? Do most of you get it? Okay, whoever said yes, sir, don't say this again. Did you guys get it? Who didn't say yes? Who doesn't get it? I mean, I'm just trying to make sure you guys get it if I say something. There is no point of me going on speaking and you'll be confused. Okay? Do you guys understand cloning? What does cloning mean? Not the dolly cloning, sheep cloning, just the cloning. Cloning means you clone a vector, a sequence in a vector. Vector is a plasmid. You guys get this. You guys have recombinant plasmid, recombinant DNA technology class, then you should get this, right? Okay. All right, we clone that. There is a promoter. You can have a target gene promoter that allows you can put this in a cell and you can again measure the level of fluorescence through anything that reads the emission from a fluorescence spectra. Okay, do you guys understand fluorescence spectra? You had your physics classes earlier. I'm sure you've had it in 12th, right? Okay, hopefully, if you are reading and not watching your YouTube videos. I keep on saying the same thing, so please don't mind. But this is essentially very simple, right? You can use your promoter of a gene. So you'll clone, let's say, 200 base pair upstream of your gene promoter. You'll fuse a GFP. You'll just go on and test whether the activity is there. Okay, that's another way to assess gene transcription in vivo inside the cell. You will just take that vector, you put it in a cell, transfect in a cell. You guys understand what transfect means? Okay, good. Okay, in a cell of any kind. It can be bacterial, it can be mammalian, it could be whatever, I don't care. Okay, I can put this in a cell and I can just measure my fluorescence after many different ways. Okay, let's not get there now because that will take up forever. You can look in a cell to see where my mRNA is. You can do that by something called in situ hybridization. You guys get this? Okay. This picture should be pretty and obvious, right? You again put a fluorescent spray label that binds to your target mRNA, and this you see the expression copies. Sometimes these mRNAs are only there in a specific organelle, right, of the cell. They're not there everywhere. And that's how you can understand whether it's a specific, it's a mitochondrial mRNA. Is it nuclear? Is it cytoplasmic? Is it whatever? Okay, get it? Okay, that's a simple way of understanding where my things are in a cell. I'm not supposed to be teaching you all of this. You should be knowing all of these things. I'm teaching you RNA expression only because it matters to epigeneticists how mRNA is expressed through my epigenetic modulation. Okay? All right. But most importantly, because we're an epigenomics course, we need to understand RNA copies at the genome level. So I care about all genes in a cell. I don't care about a gene X, right? I was lying. I don't like it, Elon Musk. I just like to look at all transcription of all genes. Okay. So RNA sequencing is used for that. Okay. We studied next generation sequencing yesterday. How many of you remember that? None of you in the back know this, right? Unfortunately. My friend, you were here yesterday, right? Do you remember next generation sequencing? Oh man, what are you guys doing in the class? Okay, I'm sorry. Gotta focus, right? Again, I am putting a lot of energy. I expect you guys to focus. Okay? How many of you do not understand next generation sequencing? My rest of my class does not understand next generation sequencing, right? Oh, this is gonna be fun. I don't think we're gonna end up on the right side of things today. कल वाले का लेक्चर का है क्या? कल वाला इसमें है क्या? नहीं है? होगा? डे 3 पीपीटी वो एक ही लेक्चर है कल का सुबह पहले वाला. हाँ, उसको लगा दीजिए. नहीं देख नहीं मैं कर लूँगा. Okay, people who are sitting in the back, can you guys see the slide? Do you read the slide? You understand English? You can read English, right? Okay, try to read the slide. Basically, what the slide says is you can take your DNA or you can take your RNA for the RNA transcription. You will convert RNA into reverse transcription. So for the people who understand this, otherwise, basically in RNA sequencing, you will reverse transcribe your RNA into DNA. Now you got the DNA, you can sequence that, right? That's RNA sequencing. For people in the back who do not understand sequencing, you can take a piece of DNA, you can break it down into small pieces, okay? So you have a long DNA, I break it down, make it small pieces. That's what on top it says, okay? Fragmentation. And then you can put two pieces of known adapters, which we call adapters. So let's say 20 base pair DNA, okay, on each side of my fragmented DNA in a process which is called library generation. You don't have to worry about the details of it, but just know that if I had this piece of DNA, I can add on two sides, I can add two pieces of DNA with a known sequence, okay? You understand known sequence? English, good? Okay, known sequence. I can design primers against those. How many of you understand PCR on the back, not the front ones? PCR, polymerase chain reaction. How many of you know what a polymerase chain reaction means? Okay, my friends, you're going to explain to the class what does PCR mean? Yeah, loudly. Yes, please go ahead. Okay. 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 Denaturation. Then next, primer annealing, elongation. Okay, that's PCR. How many of you really gone to a lab ever? How many of you done an experiment in the lab? How many of you actually have done a PCR in the lab? Okay. I'm meeting your vice chancellor today. I'm going to try to tell him to get lots of PCR machines in the lab. Okay? Because you guys, if you can't do PCR, my friend, your life is doomed. It's kidding. It's okay. You should be able to do PCRs, at least in a university setting, right? Okay. A polymerase chain reaction is something that is used to amplify a piece of DNA through repeated rounds of amplification of that DNA by use of a DNA polymerase and a DNTP. The DNA polymerase means what? Okay. DNA polymerase does not mean tag polymerase, guys. Okay. Tag polymerase is a kind of DNA polymerase. All right. People on my back, not the front ones. I know you guys know this. What is a polymerase? What does polymer mean? What does a polymerase mean? You want to tell me? Yes. What's your name? Yes. Ashwarya. What does a polymer mean? Okay, that's polymerase. Good. Why don't you guys speak up? I don't bite, as I said earlier. And none of the other friends do, right? Please. Okay, speak up, guys. Be engaged. Okay, so in a case of a sequencing reaction, those primers are against the adapters that we ligated in the first place are on a chip like that. You see, that's called flow cell. Okay, there are billions of those in those flow cells. Okay, billions of those, right? And there are lots of reactions that go on which are called a bridge amplification. So there are two primers, amplify, and it's done in the context of fluorescent nucleotides. And the fluorescent nucleotides are detected by a reader and a camera, and they take pictures, and then they really detect what sequence was it, because my A was a different fluorescently tagged, B, T was differently, C was differently, G was differently. So by looking at the sequence of images, I can tell you what my sequence was. That's a machine which is called sequencing by synthesis, SBS, Illumina. Have you guys heard of Illumina? High-seq sequencers? Those are called sequencers. They cost millions of dollars. You guys have not heard of them. Oh, it's okay. That's fine. I don't expect you to hear of it if you don't know PCR. Okay? But that's next generation sequencing. That has revolutionized the world of genomics, the revolutionized biomedical science in last 25 years. And unfortunately, a lot of you don't know this. Okay? 15 to 20 years. So we really need to put our game up now. Okay. All right.